Welcome, everyone. I think people are just slowly starting to come in, so I will do a short introduction, but I will speak slowly to give people the chance to continue joining us. So thank you for coming on the webinar today, for joining the conversation on the role of viral vectors in emerging cancer therapies. Viral vectors are an exciting technology that we don't speak enough about, so I'm thrilled to be co-hosting this session in collaboration with BioCanRx and the National Research Council of Canada. Together, these organizations are not just advancing science, they're catalyzing a shift in how we understand, develop, and deliver cutting-edge therapies to patients across Canada. In today's session, we will unpack a critical component in the cell and gene therapy landscape, which is, of course, biovectors. These biological tools are foundational to how we deliver transformative therapies like CAR-T. And to guide us through this conversation, we're honored to welcome two leaders in the field, Dr. Kelley Parato, who's the program director for the NRC's Disruptive Technology Solutions for Cell and Gene Therapy Challenge program. Kelley is a born problem solver. Throughout her career, she's been the type of person who sees challenges to be addressed rather than problems that can't be solved. How fitting then as the program director, she's now been tasked with addressing some of the key issues affecting the accessibility and affordability of cutting-edge cell and gene therapies in Canada. Kelley completed her studies at the University of Ottawa, where she obtained her PhD in biochemistry, microbiology, and immunology. Because those three were not enough before working for many years at BioCanRx. Now, after a little over a year as a valued member of the Human Health Therapeutics Research Center, she's thrilled to be a part of significant initiative for the benefit of Canadians. And Dr. Stephanie Michaud is the president and CEO of BioCanRx, a not-for-profit that seeks to accelerate the delivery of innovative immunotherapies from the bench to the bedside. In this position, Stephanie is responsible for running all facets of the organization. She brings more than 20 years of public, government, and private sector experience in research and science and technology innovation policy. She strives to create partnerships between government, not-for-profits, academia, and industry to maximize the impact of research funded by the BioCanRx network on the lives of those affected by cancer. Stephanie earned a PhD in organic chemistry from McGill University. She is involved with a number of not-for-profit organizations and is a member of the board of directors of Research Canada. Together, they'll explore not just science, but the strategy, how viral vectors are currently being used, what's needed to scale their impact, and what this means for cancer care across the country. And so just before we get started, just a few housekeeping notes. The webinar is being recorded and will be made available after the session at cancercolab.ca and on our YouTube channel at Cancer Collab. We do have time scheduled for Q&A at the end of the discussion, but please use the chat box or the Q&A box to submit your questions at any time. And so with that, I will turn it over to Kelley to kick off today's discussion.
Great. Thank you so much, Sabrina. Thank you very much for the invitation to join. It's great to get out of the lab and get out of the office, so to speak, and get to talk about something that I've always been very passionate about. Could you just give me a thumbs up if you're seeing the correct presenter view? Great. Okay, thank you so much. So I was invited to sort of give a little bit of an overview of the use of viral vectors in treating cancer. And this is actually, I know that, CAR-T is something that has been mentioned in the introduction and is certainly very prominent, and I'm definitely going to cover that. But there are actually several uses of viruses in treating cancer, and I kind of wanted to situate these various fields of application and why this works and why this is exciting. And so that's really how I tried to position the presentation before Stephanie takes us further on the journey. So with that, I sort of wanted to come to something that, that might sort of rack our brains a little bit. Viruses normally cause disease, right? Viruses are infectious agents that make us ill when we get sick. So thinking about them being useful as a research tool and as a therapeutic strategy might just seem a little odd, but I'm going to try to convince you that this is actually very smart and no longer science fiction. And so viruses, as I've said, they are infective agents. They typically consist of a nucleic acid molecule. This is the genetic information of the virus, and it's encapsulated in a protein coat. And viruses are really interesting. They're not actually alive on their own. They don't multiply by themselves. They don't consume nutrients by themselves. So they're not really independently living organisms. They are more of a parasite. In order for them to survive, to multiply and produce progeny and spread, they actually need to infect a host cell. And so I've really boiled down the life cycle of a virus to 4 simple steps. Most viruses have some version of this pathway in their life cycle. And I've done this for a reason, because this is how I'm going to reflect how these components of the life cycle can actually point to how we can use viruses instead to treat cancer. So viruses bind by virtue of a surface receptor on a host cell. That binding event causes the cell membrane to literally swallow the virus particle that's attached on the surface. And it literally, the cell will then digest the outside of the virus particle, will take away the protein coat, liberate the virus components, the genetic information into the cell. It then uses the cell's normal genetic processing machinery in the nucleus to actually create new strands of genome and new protein and new virus particles. So the cell has actually become a virus factory. And now this cell is going to spit out all the progeny virus that's been produced. So what's really important about this is that usually the cell that's been infected dies from this cycle. And usually also an immune system is created. There's usually some source of inflammation during this replication cycle. And this inflammation invites the immune system to come in and also address the virus challenge. So we're seeing this happening in a normal host cell. This could be your lung, it could be a blood cell. And that's when we get sick, when this cycle is happening in our healthy tissues, and there are consequences of our cells dying, and there are also consequences from inflammation and immunity. So, but what if we could actually orient that towards cancer and not our healthy cells? And that's been the question that I would say kind of summarizes how we think about using viruses as therapeutics, is redirecting the same functions that they have in their, I'll call it day-to-day life, but really to manipulate them to use, to infect cancer cells or become tools for other therapeutics. And that's how I've divided the talk. I'm going to give you 2 examples where, just by virtue of how a cell progresses through its life cycle, we can reorient them to cancer cells instead of healthy cells, and also how viruses can be used to transfer therapeutic payload, therapeutic genes to cancer cells, rather than the virus's usual genes, which might make us sick. And then on the right-hand side, I'm going to talk a little bit later about how viruses can be used to actually manipulate the function of a cell that it has infected for a therapeutic gain. And so, starting with that first example, viruses can be used to, can be made to infect and kill cancer cells preferentially. So discovery, now about a couple of decades ago, had revealed that there's some really critical differences between cancer cells and normal healthy cells. One of these really critical differences is in the antiviral protection mechanisms that our healthy cells have. It's called an interferon pathway. That's such that when normally a healthy cell is infected with a virus, that infection event triggers the production of a substance called interferon. And that interferon spreads to other neighboring healthy cells and actually makes those cells resistant to virus infection. So you would think that normally cells would be very protected from viruses through this means. Most viruses have evolved mechanisms to get around that protective event. But we know how viruses do this, so we can now mutate the viruses. So they're now sensitive to this antiviral infection event. And so we've actually done this with an oncolytic virus, where we've taken away its ability to hide from interferon. So if we treat an animal, if we treat a test subject that has a tumor with this mutated virus, the virus is going to infect healthy cells. It'll infect the first cell, and then it will stop there. It will go no further. The interferon response will shut down that virus replication. But cancer cells very uniformly don't have that antiviral pathway. That antiviral pathway would stand in the way of all kinds of other functions that a tumor needs to be a tumor, like creating a new blood supply, like dividing unchecked and growing without limits. And so now this virus that we've mutated that is very sensitive to an interferon response infects a cancer cell, which can make interferon, but they don't respond to the protective effects of interferon. So now these cancer cells are actually infected wildly with this virus and killed in the process of this virus infection. But neighboring healthy cells are completely left intact. So this is not science fiction, and this is not preclinical studies anymore. There are 4 examples of what we call oncolytic viruses that have been approved in various markets, one very notably in the United States several years ago for the treatment of metastatic melanoma. And to date, when I do a search on clinicaltrials.gov, there are now 216 different clinical trials worldwide using this type of cancer therapy. 57 of them were used as a single agent. But as is becoming increasingly popular with many formats of cancer therapeutics, now oncolytic viruses are being combined with other immune therapy strategies. So there were another 63 different clinical trials available that combined the two, the antiviral attack and then the boost with an antiviral immune type, so antiviral or anti-cancer immune type strategy. So that's been very successful and exciting, and there's quite a lot of activity there. And I would say a second therapeutic modality using viruses in quite a similar way is to, rather than relying on the virus's ability to kill a cancer cell, but can viruses target a cancer cell and deliver a therapeutic payload. So we actually know how in the lab to substitute some of the viral genes that would normally cause pathogenesis with therapeutic genes so that now the virus is going to go and infect a cell, is going to replicate, and instead of making pathogenic genes, they're going to make an anti-cancer gene. And so there are, as of this publication, which I admit is a few years ago, but there were over 1,000 different clinical trials ongoing using viral therapies for cancer gene delivery. And so I've got in this figure here, cancer gene delivery generally is actually a very significant field of research. There are many ways to introduce therapeutic genes into a cancer cell. You can see everything from the mechanical versions, a gene gun or a microinjector, to literally poke a gene into a cell. There are several particle formats that you can encapsulate a genome, you can coat it with genome, and actually have the cancer cells swallow them up passively. But then, of course, there are also several viral vectors that can be used, and you code the therapeutic gene inside the genetic material of the virus. So when the virus infects the cell and copies itself, it's also copying and expressing that therapeutic gene. And so several clinical trials in cancer therapy have included genes that have direct anti-tumor activity, so the gene is toxic and will kill the cell. There are genes that can disrupt a tumor blood flow, so we call them anti-angiogenic, so they can strangle off the blood flow to the tumor and sort of indirectly kill a tumor that way. Many of these studies involve immune stimulating genes, introducing something that's actually going to inflame the immune response, and then let the immune response be the therapeutic that takes over. And also, of course, enzymes that can degrade components in the tumor microenvironment that are necessary to support the survival of the cancer. And so then the last example, and a very prominent one that I think many of us are very familiar with, is the use of viruses. Instead of it being the therapeutic, it's actually as a critical ingredient to re-engineer the function of a cell that it has infected to have a different function. Just I'll say that very generally. So CAR T cells are a very important example. They fall in a class of immune cell therapy. I'll only spend one slide on immune cell therapy, which is really the premise is to either activate the immune response or engineer an immune response that doesn't already exist to direct that immune response to treat a patient's cancer. And so this can be done by taking cells that already exist, for example, in a patient that are already specific and known to target a cancer, but they need to be perhaps activated in the lab, ramped up, supersized, et cetera, grown in high numbers and given back. They naturally have an immunity to cancer cells. But then an alternative is to take any immune cell in our body and engineer it to become a cancer killing cell. And that's what engineered CAR T cells are. I'm going to explain how viruses are used. So the chimeric antigen receptor T cells, or CAR T for short, these are cells, they are derived typically from T lymphocytes in the blood of a patient. And they're taken to the T cell population is manipulated in the lab. We insert a gene that so instead of this T cell recognizing whatever it was born to do, recognizing flu, recognizing a bacteria, etc. These clones exist in our body in circulation, very passive until you find an infection. They're there at the ready in case you need them. So if we take them out in large number and we say, never mind what you were going to do, now you're all going to be specific for this cancer cell or this cancer antigen. And so we reprogram those T cells by introducing a virus that's carrying a new T cell receptor that will superimpose over the T cell receptor that the cell was born with. And so really this is how T cells are made in the lab. And the viruses are this critical engineering vector that will introduce the new DNA for that new receptor into that T cell. We grow the cells in the lab and we give them back to the patient. And now the patient is full of these engineered cells that have the activation and the specificity to go recognize that patient's cancer. So, and this is a really prominent and very important class of therapeutics. This was really game-changing in the immunotherapy space. I love to talk about this example because it was just so successful. There was a young patient who was the first pediatric patient to receive an engineered T cell. Her name is Emily Whitehead. If you've heard any of these webinars about CAR Ts before, you've probably come across her name. But back in 2010, she was diagnosed with ALL. Standard of care treatments had failed to treat and support her cancer. So by 2012, when she was deemed to be terminal, she was enrolled in a phase 1 clinical trial led by a researcher at the University of Philadelphia at the Children's Hospital of Philadelphia. And she was, as I said, the first pediatric patient to receive genetically modified T cells to fight leukemia. And in a very short period of time after her treatment, she was put into remission. And she has grown up to be a healthy young woman, an advocate for cancer research and patient awareness, et cetera. So she's a healthy, active young woman and a really critical success story for this platform. And so there's been a wave of activity for CAR T now. So 6 different products have been approved by various jurisdictions. You will see here, I'm not going to go into all these details, but I'll highlight that these are all directed at some form of a blood cancer. They are all cells that have been engineered with some sort of a virus, typically lentivirus or gamma retrovirus. 2 important categories. And some of these products vary in terms of various, I'll say, co-stimulatory domains that are part of the functional molecule that is directing the cells to recognize and kill the cancer. So that's what some of these differences are. And of course, their approval in various key indications are also discriminators for these products. And then I think my last slide really, before turning over to Stephanie, is just to talk about what's next for this field. Where is it going? I mean, this has been so promising. There are tremendous response rates and durable response rates in a very significant number of patients that are fortunate enough to receive this therapy. And so this whole cycle of manufacturing, you can probably see it's very different than how a standard chemotherapy drug would be manufactured. It's very complex. It is right now it's a personalized therapy derived from one patient to treat one patient. And so this is time consuming. It's incredibly costly. It's a very complicated supply chain. So there's quite a lot of activity now in research just across this platform, looking to move to different cell sources rather than having to use a patient's own tumor cell to treat them, that perhaps there's a way to bank large numbers of donor cells, third-party donor cells, I will say, and render them tolerable to be received in patients that are not, the same patient and not necessarily a match. And so this is what we call allogeneic or third-party donor cells as a platform for the cell. And that would be a huge improvement to manufacturing. On the viral vector side and that lab manipulation, there's quite a lot of innovation focused on making lentiviruses and other engineering viruses much faster and much cheaper because the manufacturing is very expensive and very complicated. As well, there are innovations coming in terms of rather than infecting a cell with a virus and leaving it to the virus to introduce the DNA, is actually editing the DNA very directly with gene editing technologies such as CRISPR. And there are also some upcoming strategies in the preclinical space looking at actually making these cells or engineering these cells inside the patient. So not having to take them out in the lab and mix the ingredients together and bring them back to the patient, but actually using viral vectors and others to target these genetic constructs into the cells that we want to transform into a cancer killer. So those are some really interesting innovations that are coming on that side. There's a lot of exploration into other types of cells, other types of immune cells that could be good, I'll call them chassis for these CARs. So natural killer cells or macrophages. The natural killer cells have different mechanisms for killing tumor cells. Macrophages might remodel the tumor microenvironment in favor of an immune response that might be a more natural response. And as well, quite a lot of innovation in terms of manufacturing devices to simplify all of this manufacturing and really bring that manufacturing almost to bedside rather than to an offsite factory. And then finally, a lot of innovation in terms of being able to bring this platform into solid cancer targeting. We've seen great successes in the hematologic space. There are some specific challenges to having these cells access solid tumors and being as effective in that space. So quite a lot of innovation in preclinical and clinical designs that way. And as well, combination therapies. So using other types of modalities such as checkpoint inhibitors, even oncolytic viruses that I mentioned early, in combination to have a 1-2 attack in the tumor. So that's my overview. I'd love to turn it over to Stephanie, and I think we're going to have an open question period after that. So thank you for your attention while I got really excited about viruses there. I'll stop sharing.
That was great, Kelley. Thank you. You set it up perfectly. So we're really going to change gears right now. And instead of looking at my calendar, I hope you're able to see my slides. Can somebody give me a thumbs up? Yep, perfect. Excellent. So first of all, thank you, Kelley. So as Sabrina has noted, Kelley and I used to work together, and she has done an enormous amount of work in the field of oncolytic viruses. And so it was she was really excited because, it's really a key area of research that she's worked on and contributed to enormously. So thanks, everybody. And I want to take this opportunity to thank Sabrina for the invitation to participate and be here with you today. So a little bit of a pivot with respect to the use of viral vectors. I'm going to be focusing in on CAR-Ts. And today, what we'll be exploring is health system readiness and the challenges and the opportunities that exist around CAR-T cell therapy that Kelley has just described, where viral vectors are used to produce them. And we're going to be drilling down a little bit and focusing on Canada's landscape. And you'll see from the slides that I'm going to show you for the next few minutes that I've pulled extensively from some fantastic reports that have come out from IQVIA and also from Shift Health, and also perspectives from recent webinars, that have been provided, by these same organizations with respect to health system readiness. So, and these have featured a very wide range of international experts and patient advocates. So, with respect to, all right, perfect, it advanced. With respect to CAR-T cell therapy and the system journey, so this is an overview of really what it takes in order to get one of these products to patients, and really describes more of starting from the clinical trial, going all the way to the actual product delivery of the product to the patient. But even before then, folks have been working on technologies adjacent and related to CAR-Ts since, I would say, the very late 1980s to early 90s. And it's really in the 2000s that we saw different technologies, and it was because the biotech field is advancing ever more rapidly each and every day, where we saw some active developments where this type of technology started to get on the radar, and because individuals were starting to understand the promise of this technology, and then were actually beginning to test what this was going to look like, and how, and evaluate how it was going to work in patients. So it's been a long time. Getting research from discovery to patients always takes a very long time. But on this slide here, we have, we're starting off from the discoveries have happened. We know that we have something that works well. This has been validated through research, through publications. It has been the work of a lot of academic researchers that have brought these different types of products to a clinical trial and demonstrated safety, demonstrated efficacy. And we're talking about CAR T, so in certain cases, absolutely astounding results. And this type of work that's required is typically termed preclinical to clinical development, and is, as I've mentioned, to demonstrate the safety and efficacy of the product. Once that type of data through clinical trials is collected, also information on the manufacturing, a very comprehensive dossier is submitted to the regulatory agencies. So in our case, Health Canada, but of course we're aware this is a therapy that's used worldwide. So the FDA and EMA, which is the European regulatory agency, and they evaluate whether or not this is a therapy that they are going to authorize for use for patients in their given jurisdictions. And this is where the regulatory authorities review this. The next stage, once a marketing authorization is obtained, is the setting of the price. And folks on the call may be aware of HTA, or Health Technology Assessment, which is when the determination of the pricing of a certain drugs is determined and recommendations are made. And then this is where countries, jurisdictions enter into negotiation with the pharmaceutical companies that are bringing these really important products forward, and with an eye, of course, to being able to provide this to patients. Potential CAR T centers, because as Kelley has noted, this is a complex therapy, and we'll get into breaking down the different steps that are involved in actually delivering this therapy to patients. The centers are evaluated that are going to be delivering this type of therapy are evaluated by the pharmaceutical manufacturer, and the criteria are listed here. So the type of infrastructure that they have available, there's training that is provided to the hospital staff. There are very specific side effects, for example, which we'll touch on a little bit later, that at times have to be dealt with, and so a certain level of experience and expertise is provided, and all of this falls under this CAR T center onboarding. So once they are accredited, then they enter into the agreements and they become a site that is able to deliver this type of product. So that is the journey for a CAR T cell to go through marketing authorization, pricing, reimbursement, negotiation, and then the accreditation of the different sites to get the product over to the patient, and really speaks about the system. But what does the patient's journey look like? So from the patient perspective, the path to a CAR T is long. It can be uncertain. I would say at times deeply personal. Patients often face fragmented referrals, long wait times, travel burdens, limited information. And as one of the panelists had noticed in the April 2025 Health System Readiness webinar, is that the gaps that exist for the patient aren't just inconvenient, they're life-altering. So if we look at the upper left-hand side of the screen here, patients are, of course, evaluated. In Canada, CAR Ts are not available as the first line of treatment. It's if a treatment has failed a patient, then patients are referred if they're eligible to receive this kind of therapy, and then a decision is made whether or not they will embark on this journey to receive a CAR T. So they're referred by their oncologist to a center that is able to deliver this kind of therapy. Initial discussions, of course, because once this therapy is delivered to a patient, there are specific eligibility requirements for the patient, not just for the type of cancer that they have, but whether or not they're going to be able to stay at the center for a period of up to 30 days once they've received their CAR T. So there are a lot of other considerations that don't necessarily arise when we're administering other types of therapies. So as Kelley has mentioned, the production of a CAR T, it's considered to be complex manufacturing. We're talking about live cells, and the starting material for the product is the patient's own cells. So the patient has to undergo a process called apheresis, and that enables us to collect the T cells from the patients, which will then be genetically modified by one of those viral vectors that Kelley was describing in order to modify the T cell to supercharge it to fight the cancer. So this happens in a parallel track. That patient then may have to undergo bridging therapy. And will definitely, before receiving their CAR T cells, once these have been modified and fully manufactured and released by the pharmaceutical manufacturer, receive a lymphodepleting chemotherapy regimen, essentially to make room for the new cells that are going to be administered to the patient. Right here is that short-term monitoring, and where patients are very closely monitored for potential toxicities due to the administration of the CAR T cell. These are T cells, so these are part of our defense mechanism in the human body to fight infection. And what we've done here is really supercharged the T cells. So the possibility of your immune system getting overreactive definitely exists, but there are tools and specific protocols that through the training administered by the pharmaceutical companies to the physicians at these specialized centers are able to deliver to the patients. There is long-term follow-up currently in Canada, and I believe in other jurisdictions as well. Once a patient has received a CAR T, the follow-up is 15 years, and this too becomes it's a lot to ask of a patient to commit right to this 15-year follow-up following the administration of the CAR T, because it means that they have to travel back to that specialized center. So there is a lot going on with respect to administration of the CAR Ts, not just from a system perspective, but also, of course, for the patient, their families, and what it means really to engage down this path of moving towards a CAR T. So CAR T access in Canada, and this is where we're going to be drilling down a little bit on what these are. Canada, as you can see from this slide, has very unique challenges. And so despite having a relatively fast referral to treatment time, a smaller percentage of referred patients actually receive the treatment. Why? The challenge really lies in what happens after referral. There is limited site capacity. There are workforce gaps, which won't come as a surprise, travel burdens, which I've touched on, and also the complexity with respect to the reimbursement process, which still exists. Other issues include under-resourced ancillary services. So for example, imaging, for example, of course, apheresis, which is a critical part of being able to produce a CAR T. There's also a challenge of uneven provider and patient awareness, especially in rural areas. So most of the centers that are accredited by the pharma manufacturers typically are in very large urban centers. And again, we also have the issue of poor data sharing, which writ large exists across Canada, which I know many different arms of government are working on, but it's still a challenge at this time with respect to the delivery of this therapy. So referring physicians cite, I'll focus on the top three major concerns with respect to the barriers, right, that are reported by these providers. And the first is patient health and eligibility. And it is because there are some long wait times and there is deterioration in the health status of the patients. Time to treatment is also an issue. And as I've mentioned, we don't have a very large number of centers across the country, and so there are definite geographic barriers to accessing these certified centers. There's also variability in knowledge and comfort with respect to the CAR-T referral process, again, especially outside of large academic hospitals. So this data here was pulled from the IQVIA Institute for one of the reports that I cited at the very beginning. And for those that are interested, this is filled with some incredible data with respect to the implementation of this technology in Canada and in other jurisdictions, the title of which is on the lower left-hand side of the slide. But if we take a look at this comparative data in DLBCL, which is diffused large B-cell lymphoma, and use this as a case study, we see that while Canada performs somewhat well in speed from referral to treatment, the overall conversion rate from referral to therapy, so actually receiving it, remains lower than in peer countries. And it points really to a bottleneck at the system level beyond patient eligibility that really deserves targeted interventions. And so part of the discussions that manufacturers are of course having is the need, and this is taking place not just in Canada, but around the world, the need for more centers so that we can enable greater delivery. So really upping the game with respect to health system readiness. So I do have another slide to show you here. One sec. Perfect. So if we take a step back and really take a look at where Canada stands, and Canada here is in this second column, we can get an evaluation of the relative levels of readiness. And it's not just about the individual centers. It's really about having a national approach to building in readiness. So some countries like Spain and France have implemented policies and reimbursement frameworks that support broader CAR-T access, but Canada is still lagging a little bit behind on these readiness indicators, particularly around equitable access and payer coordinations. So some of the recommendations that have come out for Canada, on how to bridge the gaps that exist, is to increase certified treatment centers, which I've already mentioned, also with adequate workforce support, ensuring equitable access for rural and underserved populations, investing in domestic manufacturing to shorten turnaround times. And for those of you that are aware of the work of BioCanRx, this is something that my organization is very committed to, and harmonizing data and reimbursement policies across provinces. And there's no quick fix, but they're achievable, right, with the right governments and series of investments. And there's so much work being done in the area that I really do believe that we're going to see a greater number of these types of therapies, and we will have to be ready because patients are asking for them. So health system readiness is not just about capacity, it's about coordination. So as mentioned, sustainable funding models for centers and follow-up care, workforce planning, clear referral roadmap for community physicians, data systems and measures of outcomes, patient experience, quality of life, not just costs, are absolutely necessary. And with that, I thank Sabrina for the invite again, and I look forward to the discussion. Thank you.
Stephanie, I wonder if you might talk a little bit about, and I hope you can talk about it,
Yeah.
but just a little bit about the work that BioCanRx is doing in the viral vectors, and maybe a little bit about what you're doing with, I believe it's Denmark.
Yes, absolutely. Happy to. So and this, it's a little bit of a background story, really speaking about the birth of the CAR T program at BioCanRx. And it was really born out of a lot of frustration because it was at a time when Canada did not have any marketed products in CAR T or anything like that. And so the discussion for us started in 2016. It was after a talk an amazing talk at one of our conferences. It was a summit in 2016 in Halifax. We had just heard an amazing talk from a researcher at Vanderbilt, and people's minds were absolutely blown away. This researcher was manufacturing a plethora of different CAR Ts, and our researchers came out both excited and a little bit depressed because the thinking was, even if we wanted to, we couldn't make one of these products in Canada because we don't have the manufacturing infrastructure. And so, as it often goes, the around a few beers, the researchers, and Kelley was part of this discussion at the time, and they decided to let's make that happen. Let's onboard some manufacturing in Canada so that we can enable research and the production of these different types of products here in Canada. And so, the researchers, the clinicians, really took the bull by the horns and decided to onboard this. It took us 2 years in order to working with a number of different partners, OICR, and a number of other groups, the NRC, also, in terms of making that happen. And we manufactured our first CAR T 2 years after that. And during that same period of time, we developed a clinical trial protocol that was informed by patients so that when we onboarded the manufacturing, we were ready to go knock on the door of Health Canada to submit a clinical trial application. And we've been treating patients ever since, and that started in 2019. By that time, Kymriah was available in Canada, although for a limited patient population, as everybody here knows. So for those of you that don't, in certain indications, certain blood cancer indications, the product was only available for those between the age of 2 and 25. That was for ALL, for leukemia. And so our clinical trials was launched to treat a number of patients that otherwise would not have had access to this therapy. And so we're still running that trial and also experimenting with making this product under the umbrella of a single clinical trial, but with multiple manufacturing sites. Why, you ask? Because Canada is a very large country, and being able to have backups is really important to us. And another piece that we believe in that could perhaps change that pathway that I spent time describing is limiting the time by being able to provide point of care manufacturing at different hospital sites. So last bit on Denmark. So we figured all of this out. We have the viral vector that's been manufactured at the Ottawa Hospital, and it's a GMP viral vector for CD19 CAR T. And Denmark contacted the researchers and the clinicians. And so that viral vector has been sent to Denmark. All of the SOPs, which are the instructions for making these different products, have been sent to Denmark, and they have run with it and are treating both pediatric patients and adult patients with the viral vector that comes from Canada. So it's a huge success story where we've been able to share the know-how, that was acquired during, onboarding of this manufacturing in Canada, and share it with other countries so that they can get their own programs up and running. And Kelley was a huge part of that.
Kelley,
And
do you
still
want to
is,
add?
and still is with other products now. So
There you go.
yeah.
Sorry,
Kelley,
Sabrina,
did you want to add to that?
No, I mean, I love Stephanie's version of that story and how it all came about, and the success of it is just incredible, right? And I think that Canada has been able to do this because of the unique way that we collaborate across our immense and diverse geography, and across our provinces and territories. And I'm hearing that, so many others are stepping up and wanting to be part of this network and have that capacity within their province. When I came to NRC, I'm no longer the lead of the Cell and Gene Therapy Challenge Program. I've shifted into a role now more in biomanufacturing and focusing on biomanufacturing innovation to help propel and accelerate cost-effective products like lentivirus and solve some of those manufacturing bottlenecks. But the dream really was to look at how could public engagement, so government engagement, actually facilitate this. So the NRC has capabilities in design for molecules for CARs, particularly the binding parts that recognize tumors. And so we've contributed a binder that we've developed in collaboration with the BioCanRx network. And so that's going to be a follow-on clinical trial product, and that'll be a made in Canada originator molecule. And again, we'd like it to be made accessible in the same way, right? So good news for us is that there has been this diffusion of benefit to Canada. So it's something that I want to see quite a lot more of.
And I'd like to And I like to use the expression, you build it and they will come. And, that comment that had been made by, the researcher whose name I forget now, like, even if we wanted to make it, we had a construct, a new CAR T to make, we wouldn't be able to make it in Canada. We can now. Mm-hmm. We can make it. And our researchers are designing new CAR Ts. So the CD22 from the NRC, and we have others too that have been tested in clinically tested. So, and using, what has been the tracks that have been laid down. So it's great to see that addition of different options for patients, in addition to what we see coming with, the marketed products.
Yeah. And just to build on that point, Stephanie, I mean, the ability to have the clinical capacity and the manufacturing capacity domestically means our own innovations are not following a cycle of getting to preclinical evidence, and then the intellectual property is purchased and developed elsewhere in other jurisdictions. And then we can buy back market products under conditions that someone else has decided to market those products. And of course, there's a role for that. I don't want to say that that pharma doesn't have a really important role to play, but so too does the domestic innovation ecosystem, and allowing patients domestically to have access to the innovation that is coming out of our university labs and our small to medium enterprises that have all these fantastic ideas, novel cancer targets, new and improved molecules that are much more effective and safe. So we'd love to be able to, we love seeing that this innovation ecosystem is really blooming now.
Mm-hmm. It's a representation, and something that we feel very strongly about at BioCanRx is that Canadian patients should have access to the best science that Canadian research has to offer, period. And a clinical trial and being able to support our own homegrown clinical trials is absolutely essential.
And I stand with both of you in support of domestic innovation. So whatever we could do in the advocacy space to help further that, I'm here for it, especially, given what is happening south of the border. I think like domestic innovation is even more important now than it has ever been.
Agreed.
Yeah. And so maybe we could talk a little bit about because I feel like none of this could be really successful without that cross-sector collaboration. And so can we talk about that a little bit?
Yeah, sure thing. So a great example, going back to the CD22, that binder molecule is from the NRC, right? It is their product. They are the ones that ideated this. The collaboration has happened, via BioCanRx, where we have supported the preclinical development of this product. And the network at BioCanRx supports biomanufacturing, but also supports that really important piece, which is the clinical evaluation of these different types of products. And so here we see an asset that comes from our own government's laboratories, right? So the public sector in Canada collaborating with academia, with hospitals, research institutes, and not-for-profit like BioCanRx to make that happen for patients.
Mm-hmm. Yeah, and what I'll say is, I mean, maybe I'm putting in a bit of a plug for NRC, but we exist. My research center is the Human Health Therapeutics Research Center, and while we have capacity to develop new therapeutic molecules that we ideate, we typically do this in consultation. Right? There was a pull. There was a need. We had a list of capabilities, and it was through consultation that many hands were raised and nominated CD22. So that's the molecule that came forward. And we, speaking, maybe I'm speaking too much on over the voices of my organization, but we really are about enabling innovation, addressing unmet medical need and challenges in our healthcare market, access to technologies. So we are much more tool and platform builders. So the CD22 molecule is an example where we built all kinds of tools and capabilities around CAR T innovation and the design of molecules, the design of viral vectors, and the biomanufacturing of viral vectors. And we needed an exemplar project to show this all works, very much like the CAR T Network's first CAR T project. We've built it, and now here's where I'm going to demonstrate that it works. So for us, that was the CD22 as well. And so now we have all these tools and platforms so that when new ideas come forward, when it's time to move from those early preclinical studies into something that's going to get closer to clinic and into manufacturing, we now have a lot of, analytics and robust methods, et cetera, for product evaluation and the manufacturing as well. And we're working, again, still with the BioCanRx network on platforms to transform the way that we manufacture the lentivirus vectors, because right now it's done primarily in adherent cell culture, and really all that means is that you're limited by the square inches in your tissue culture dish or in your vessels to how much you can actually make. But we've at NRC we've got cell lines that are owned by the Crown that actually grow in suspension culture, so they can grow to much higher density, and it's a transferable technology to many industry biomanufacturing labs as well. So that's again an area where we see ourselves as trying to propel new capabilities and then make them on offer and distribute this capability or work collaboratively with groups that are bringing forward the next therapeutic platforms and molecules.
Yeah, and it's really important because sorry, Sabrina, but because the cost of goods in manufacturing these products is almost always increasing. So, these types of innovations to reduce the cost are absolutely critical.
I just want to ask a couple of questions that are in the Q&A. So thank you for your patience on this. Do pharmaceutical companies handle the cell culture at CAR T centers, or do they just provide the viral vector and process training? Can a CAR T center administrative multiple CAR T products or committed to one product only?
I think that they can administer a variety of different CAR T products. The role of the CAR T center is to receive the patient and then undergo the process of apheresis. And then the cells from the patients are then sent to the pharmaceutical company's manufacturing center, and they have complete control over the viral vector that they're using and also the process that they're using. They then manufacture the product, they release it according to the specifications, right, that they've communicated to the regulator that falls under their marketing authorization, and then it's sent back for administration to the patient.
Yeah, and I think like just keeping on this, because BioCanRx is manufacturing viral vector, you could potentially, because I know Gilead is the only one who has their own viral vector. So if anybody, like any pharmaceutical company is manufacturing CAR T or anything that needs a viral vector, potentially it could be like a Canada first approach where the company is utilizing BioCanRx's viral vector to administer.
So they have their own viral vectors. They're all different. And this is, for these types of products, these take a very long time to develop. And once you have submitted your information to a regulator like Health Canada, it's extraordinarily costly and difficult to change. right?
Yeah.
So once they have locked down, this is the viral vector that we're going to use for manufacturing, they would go to a qualified, if they're not making their own, they would go to a contract manufacturing organization that has met the requirements of the regulator and obtain it, either have that qualification themselves or else get it from the contract manufacturer themselves. So, and any change to that is very costly and requires a lot of engagement with the regulator, and something that you don't want to do once you're far ahead and you've been producing a product and you have clinical data to support your submission to the regulator. You don't want to make any kind of change to your manufacturing at all.
Right, but the only one who actually manufactures their own viral vector is Gilead. And so if a company wanted to submit to Health Canada, I mean, let's say Pfizer comes up with a CAR T in like 5 years from now, they could potentially use BioCanRx's viral vector to do that.
Sure, they could if they wanted to. Sure, they could, but
Unlikely.
what we only manufacture for early stage clinical trials, and so once you get into commercial products, it's a different level. So you have to have, of course, right, a specific license, et cetera, et cetera. So, and maybe I'm getting too much into the weeds.
Yeah. Okay, I've got another question. What strategies can be used to overcome the immunosuppressive tumor microenvironment that poses a significant challenge in the CAR T approach in solid tumors?
I can jump on that one. This is a challenge for any type of immunotherapeutic strategy. Tumors have a lot of ways to put up defense mechanisms to the immune response, because our immune system technically can recognize tumors all on its own, but the tumors do tend to block the penetration by infiltrating immune cells, or their activation when they do arrive. So that they can't execute their killer effector function. And so there's a lot of science understanding the variety of ways that tumors do this. And so very often combination strategies in immunotherapy are very important, right? So I talked about early on oncolytic viruses being used as combination strategies. Working with, for example, immune checkpoint inhibitor blockade. So there are some very important monoclonal antibodies to some of those breaks, I'll call them, that the tumors put onto the immune system. You can actually interfere with those with antibody therapeutic products. So block the immunosuppression, and then your CAR T could be effective. That would be the same for an adoptive transfer, a vaccination type strategy, et cetera. So I know that those are very general. So the immune checkpoint inhibitors are quite promising. There are also thoughts around generating CAR Ts that not only have the tumor specificity, but also carry potent immune stimulating cytokines with them to counteract immune suppressing cytokines that might be in the tumor microenvironment. So those are just a few examples.
I've got another question. A 15-year follow-up requirement was mentioned for CAR T. What ongoing research is being done to explore potential biomarkers, or perhaps using digital health tools that could streamline or personalize long-term monitoring? Maybe we can make this process shorter. And actually, I would like to add on to that question. And I mean, 15 years is a long time, but there must be a lot of data that is generated from that that could also be used.
100%. And so, and that this is subject matter under active discussion, because yes, of course, telehealth, asking the question, does that patient really need to go back to, that center where they've received the therapy? Can we use their local doctors, their local oncologists, to enable them to be able to do this? Can we use telehealth and enable the follow-up of these patients? These are all subject of active discussion and solutions that are being proposed. So to be determined, but hopefully, of course, it makes absolute sense for it to be this way. And so, but yes, I know that there's ongoing dialogue on this subject, of course, because it just makes sense, right? And I think as these therapies, our healthcare systems become more habituated to and knowledgeable on these therapies. and they're deployed in a wider and broader manner. I think the experience and comfort level, across our physicians, our institutions, our patients, will undoubtedly increase. So I think there's change there, but unfortunately, yes, that's still the way forward right now. But again, subject of active discussion.
Yeah. And I'll add on to that. It's not my field, but the medical devices field is actually advancing quite a lot of, personal surveillance type equipment that can be leveraged for remote trial participation and monitoring. So that is a very consistent challenge, I will say, beyond the CAR T space. Yes, the 15-year follow-up is a particularly high bar. But I would say also enabling participation in rare disease clinical trials, right? Where the patient might be that much further from the site where they need to be. So it is a hot topic, and there's definitely innovation in the medical device area for wearable bio devices and biomonitoring.
Yeah. Justin has another question, but Justin, I will ask it offline to Stephanie, and we'll make sure to get a response for you, because it is 1:00, and I just want to, maybe we can just close off. I want to ask you, if we look 5 to 10 years ahead, what's your vision for viral vectors, and how could it change the standard of care for cancer patients in Canada, and what does success look like? So, quickly, maybe just a minute each.
Yeah. I think in 10 years' time, we will be producing viral vectors with much greater precision and cost effectiveness, smaller scales, greater output, greater safety, really rapid development. I think we're going to have tools for that. I'll leave it there because I'll only go to 10 years ahead.
No, I completely agree
Yeah.
with Kelley. The pace of change it's unbelievable. Kelley mentioned during her presentation, developments that are taking place with respect to in vivo generated CAR Ts, right? Where we wouldn't have that apheresis step, shorter manufacturing times, and ensuring that we have the right tests and technologies to say, yes, the product is good and can be administered to patients and then expanded in the patient. All of these are definitely on the horizon, and I think we're going to see great developments in the next 5 to 10.
Wonderful. I'm excited to see what happens in the next 5 to 10, and probably less than that as well. But thank you so much, Stephanie and Kelley, for your time on a Friday afternoon. We appreciate it so much. Of course, the webinar will be recorded. It will be up on cancercollab.ca and on our YouTube. So thank you, everybody, for coming in today. Have a great weekend. Thank you so much.
Thank you, Sabrina.
Thank you so much. Bye.
Bye.